In this three-part series of videos, we're going to be learning about genetic regulatory structures called operons, which is actually a long sequence of multiple elements of DNA that are used to regulate transcription at the genetic level. Now, this might come as a surprise, but operons are not actually found in human DNA. They are specific to prokaryotic genomes, meaning they only exist in the genomes of organisms like bacteria, but we study them nevertheless because they serve as a useful model to understanding how more complicated eukaryotic genomes regulate their transcription by comparison to prokaryotic genomes, which are many times relatively simpler. Now, if we take a look at the structure of an operon, we see that there are a number of similarities to human DNA in that we have the primary regulatory sequence of DNA upstream of the genes, which is what we call the promoter, and this is where RNA polymerase binds, opens up the DNA, and begins transcribing genes. Now, what makes an operon distinct from what we see in eukaryotic organisms is that there are actually multiple genes, meaning multiple proteins are produced, and these proteins productions are all regulated by the same promoter. This kind of structure doesn't occur in eukaryotic genomes. Typically, eukaryotic genes are each regulated individually by a single promoter, but what prokaryotic genomes do is that they very often have multiple proteins that all serve related functions to each other, regulated by a single promoter. Now, downstream of the promoter is another regulatory sequence called an operator, and this is where the name operon comes from. Now, rather than RNA polymerase binding to the promoter, there is a specific family of proteins that are what we call transcription factors that will bind to regulatory sequences such as an operator, and depending on what these transcription factors do and how they bind to the operator, it could have one of two possible effects. Number one, a transcription factor might actually make it easier for RNA polymerase to attach to the promoter region, and if this is the case, that means that more transcription of the operon's genes is going to occur. However, what we're going to be looking at in these three videos is the situation where a transcription factor that binds to the operator actually makes it harder for RNA polymerase to bind to the promoter, usually by blocking it outright and therefore suppressing transcription entirely. In fact, that's the very situation that we're going to talk about now, are specific transcription factors that specifically prevent RNA polymerase from binding to the promoter at all, which are what we call repressor proteins. And here we can see that the repressor protein has bound to the operator, and we see that its structure actually overlaps with the promoter sequence here, which means that if RNA polymerase is coming in to bind to the promoter, because the operator is blocking the way, either RNA polymerase won't be able to bind to the promoter at all, or it simply won't be able to get past the repressor and begin the transcription of genes as long as the repressor is in the way. Now, naturally, it's important to regulate the repressor, and most prokaryotic genomes actually have multiple ways to regulate the repressor. So the first thing that we see here is that because the repressor is actually a protein, that means that the repressor is controlled by a specific gene with its own promoter. So we see that here the repressor gene is used to produce mRNA, and then after translation, that is used to make the specific repressor protein, which can then bind to the operator, and production of the repressor protein itself is independent of the operon that it regulates entirely, meaning that its production is usually controlled by some sort of environmental factors outside of the operon itself. Now, at the protein-specific level, we can see here that the operon, the repressor, in addition to having an active site that allows it to recognize the DNA of the operator, repressors usually also have some sort of allosteric active site that allows specific substrates to bind and therefore cause conformational changes in the repressor's structure. Now, what these conformational changes do is going to affect the activity of the repressor protein and change its ability to bind to the operator and therefore change its ability to influence transcription. For example, of a substrate that can bind to the allosteric site, one possible substrate is what we call inducers, and when an inducer binds to the allosteric site of a repressor protein, these actually reduce the repressor's ability to bind to the operator sequence, causing the repressor protein to actually fall off of the operator's genetic sequence, therefore resulting in an increase in transcription, because if the repressor protein is no longer bound to the operator, RNA polymerase is not able to get in the way, or rather RNA polymerase is not obstructed and is able to begin transcription completely uninhibited as normal. Now, the opposite situation is when we have a compound that we call a co-repressor. So co-repressors actually increase the repressor's ability to bind to the operator sequence, or specifically, they cause a conformational change that activates the blocking ability of the repressor and therefore make it more likely that the repressor is going to be able to bind to the operator sequence, resulting in a decrease of transcription, because if the operator is blocked by the repressor, RNA polymerase has a much less likely of a chance to be able to get at the operon genes and begin the process of transcription. Now that you have a foundational background about the components of an operon, in the next video we're going to take a look at the first example of operon, which is the sequence of DNA that specifically regulates lactose metabolism in certain bacteria.